Bismillahirrahmanirrahim. Assalamualaikum, class. So we meet again for another topic on DDA, right? So for today's topic, we're gonna look at a little bit on a pulmonary drug delivery, okay, in terms of aerosols technology, right? Okay. So today's topic will be pulmonary drug delivery, pharmaceutical chemistry, and aerosol technology. So for today class, we will look at about a little bit on aerosol technology, disease therapy, formulation variable, and some future developments. So a little bit history on aerosol technology in DDA. So in 1980s, a propellant-driven method dose inhaler (PMDI) has revolutionized asthma therapy. PMDI is the some of the most common aerosol therapy for asthma that we know. We call it inhalers that we use, okay, spraying that were common and can be seen commercially. And then we have DPI or dry powder inhaler, which very primitive, somewhat ineffective system that deservedly did not compete with MDS inhaler in terms of physician and patient acceptance. And then in 1990s, okay, non-ozoning depleting propellant system appeared to replace CFC. I think CFC chlorofluorocarbon. Chlorofluorocarbon has some green effects or ozone effects, okay, of its use. So it will be replaced for other system extending the application of MDI technology. We can say that PMDI and the DBI, dry powder and propellant-driven mid-dose inhaler, were quite similar. However, the efficiency is very much different. It both use aerosol technology. However, the differences is in terms of physician and patient acceptance. That's why nowadays we a little bit difficult to see DPI used by patient. More or less, we can see the use of PMDI, okay, in terms of application towards commercialization and also towards the application of the patient. So, right, the deposition of drugs in terms of aerosol in the airways to the lungs is dependent on four factors. Okay, we know that we have different stages of lungs towards uh, showing that nasal cavity, bronchus, and bronchioles. Okay, and then the first that relates the parameters that relate to towards the deposition of drug. First is the physicochemical properties of the drug, the formulation, right, in terms of excipients, in terms of stability, and in terms of uh, solvent use and so on. Okay, the formulation in the system, the delivery on how effective this kind of instrumentation delivered the drugs, the way or mechanisms of this type of aerosol, different type of aerosol that have been used to deliver the drugs. And the fourth one is the patient itself, breathing patterns and clinical status of the patient. So a little bit on, we look at aerosol technology. The device that employed to deliver drugs to the lung may be divided into three categories. So basically, these three categories are main categories, okay, in aerosol technology. So MDIs, we know the propellant, and then DPI, dry, and then the third one, okay, is nebulizer. Okay, MDI is inhaler commercially with well-known, well-used, okay, we can see in terms of used by the patient. And then the third one is nebulizer. Nebulizer, you can say the use of gases. Okay, so we will look at it some example later, but we can say the nebulizer is quite big, right? It's the mobility is less compared to MDI, so we can say the MDI. That's why the MDI is more prominent towards the patient. Okay, but compared to DPI have a problem in terms of efficiency. So that's why we can say a lot about MDIs and not DPIs. Okay, so containing MDIs containing a mixture of propellant and drug under pressure. DPI utilizing the patient inspiratory effort to disperse medication, and then nebulizer using compressed gas or the vibration of piezoelectric crystal to aerosolize liquid. Okay, so the aerosol technology in terms of particle preparation, in terms of either it is the use of nebulizer, the use of MDI or DPI, the particle preparation of the drug is, can say, is very important. It is because the prepared in size, range, and width structure suitable for delivery to the lungs. So that's why the particle preparation is one of the most important part in the aerosol technology. Okay, most common method in particle size reduction is by conventional crystallization or either precipitation followed by drying techniques. So the common use, okay, been used technique for particle preparation is spray drying, which involve forcing the drug solution through a nozzle at high pressure into drying a stream from which small particles can be recovered that are suitable for inclusion in aerosol product. Right. So this is important. All right. This is important. Spray drying schematic diagram. Again, this is important part in terms of schematic diagram of particle preparation in terms of spray drying. So we have feed tank, drying chamber, cyclone, right? The feed tank will feed the feed, right? And then drying chamber, and then the product will produce a reduced size of particle. Okay, this is some kind of important schematic diagram in particle preparation. Other than spray drying, we also use spray particle fluid. However, it's quite difficult to handle this kind of implementation or instrumentation in particle preparation. So that's why the idea of spray drying, the use of spray drying is widely used compared to spray particle fluid. Right. In terms of, we look at a little bit on every part of uh, the article. Oh, sorry, every part of the aerosol system, the technology. First, we look at the PMDI. Okay, drug delivered from PMDA are initially prepared as suspension or solution in selected propellant. So the drug will be the formulation of the drug in PMDI will be in a solvent system or suspension system, right? Other components are added to aid in suspension of particle of drug deposition into solution. So other excipient, okay, are added towards the solution that contain the drug in order for mainly for good dispersion within this whole system. These additive or excipient may be co-solvent other than solvent such as ethanol or some kind of surfactant such as oleic acid. And the original, in the early days, PMDA employs CFC, right? However, due to, right, then it will have been replaced with hydrofluoroalkane or HFA due to the associated atmospheric ozone depletion associated to CFC. So I think this issue is quite known, quite been debate been long time about the use of CFC. So nowadays it been used in PMDI. HFA has been replaced. Okay, to replace CFC. Right. So we look a little bit on principle of aerosol delivery from PMDIs, right? Right, this is the schematic diagram of the PMDI that we use. I think all of you where how it can be used, okay, well known, right? So how the PMDI mechanism work? So a small volume of homogeneous diffusion of the drug in solution or suspension in high vapor pressure propellant or propellant blend from a reservoir is isolated. So meaning the solution were homogeneous dispersed with high pressure of HFA. So next, the small volume container, the metering chamber is open to the actuator nozzle. So in terms of here, right, chamber will be open, and then the difference between the difference of the atmospheric pressure within the inner and the outer segments of the whole system, okay, will uh, make sure that all the system will come out, will emit the drug system. Alright. So next, the metering chamber filling and opening to atmosphere are achieved by means of metering valve. Once opened to the atmosphere, the high vapor pressure within that atmosphere, within that system, okay, in the container of the metering valve immediately begin to equilibrate with atmospheric pressure. This has effect of propelling the contents rapidly through a nozzle, which causes shear and droplet formation. Throughout this process, the propellant is evaporating, propelling, sharing, and ultimately reducing the size of droplet produced. Right. So, the inner, right. So, the pressure within this system and the atmospheric pressure is different. Like we can say the imagination of concentrated. low concentration, high concentration. So high concentration, we go to low concentration drug. So we can say it almost the same. The high pressure here will will put out, will emit the whole system here. All right, this is schematic diagram of propellant driven method, PMDI. We have container, valve system, actuator, all right, suspension or solution in propellant, and then metering chamber, right? So in DPIs, okay, DPIs quite difference in DPI, all right? Powder product a blend of respirable drug particles and large lactose carrier particles. So in DPIs, they use lactose. Okay, they use lactose. Early design employ a unit dose gelatin capsule metering system and multiple unit dose blister discs and roll or reservoir powder devices. We have different type of DPI here. The example of blister, rolls, and or reservoir powder devices. A variety of mechanisms for assisting with the dispersion of the powder have been adopted, including impellers, compressed air, vibration, and impact hammer. So these are mechanisms on how the powder will be dispersed within the lactose. Okay, this is type of DPI, and then this how mechanism on how the drugs were um, dispersed in the lactose system, right? So here how the general principle of the powder delivery in DPI. A powder bed is exposed to shearing air, okay, that entrance particle. A blend will employ the fluidizing effect of large lactose particle to help disperse the respirable particle associated with their surfaces. The small drug particle will be carried to the lung of the patient, while the light carrier particle will be deposited in the mouthpiece of the inhaler or the aerobatic of the patient. So that's why the differences in DPI, MDPI, because the diffusion or this dispersion of the particle molecule is quite different. They have small particles. They also produce a large particle in DPI. Okay, it's not the same for compared to PMDI. How that's the problem in DPI. That's why the efficiency is less compared to PMDI. So because we have small drug that will carry into the lungs, meanwhile the bigger particle drug will be stay in the mouth. So that's why the efficiency is less, right? So here's an example, the comparison between the DPI and PMDI. So this is common, we use, and then we have here the common the DDs type of DPI. All right, the differences between metered dose inhaler and dry powder inhaler. So next, we look at nebulizer. So nebulizer employ energy from compressed gas or piezoelectric ceramic to generate droplets of water containing drug. The principle of a jet dispersion. Okay, how it's work? The drug solution is drawn from a reservoir through a capillary tube from by the venturi effect. Okay, in principle, a low pressure region is created at the exit of the capillary tube when compressed gas is passed at high velocity over the tube, throwing liquid into the air where droplet are forms. So we can say it almost the same. In terms, the third, all of these mechanism were based on aerosol technology, on the idea of how aerosols work. So more or less, it almost the same. However, the system is different, either NDPI, DPI, or nebulizer. So this is example of nebulizer. Okay. Next, large droplets are projected onto a baffle where they are located, and small ones pass around the baffle and are delivered to the patient lung on their respiratory flow. So we can see here it's quite difficult in terms of mobility compared to common inhaler. So that's why we usually see the common inhaler, okay, which in PMDI compared to nebulizer. All right. So we look at a little bit on the disease therapy of aerosol technology. So usually it been used to treat asthma, the problem that related towards the lung, the respiratory, right? So that's why we look at we enhance the disease therapy towards the lungs problem. Okay, so first is the agonist BAAs, okay, drugs to treat asthma. Aerosol BA must be delivered to the periphery of the lung to produce their pharmacodynamic effect. Very short duration of action in the lung following the delivery of BAA. So medication that relax this BAA relax muscle of the airways, which widen the airways and result in easier breathing. They are distributed in the lung and periphery, so must be delivered to the periphery to produce their effect. So drugs with longer duration, locally metabolized, and reduced toxicity were developed. Okay. Next, this is was BAs is most common inhalers use. So and then we have anticholinergics. Okay, anticholinergic acts on the central airways, the location of the majority of the receptor. So apatrobium (BI) was the first of this agent. Okay, this time of anticholinergic agent to be approved, followed by oxytropium. The success of triotropium in the treatment of chronic obstructive pulmonary disease, for which there had previously been no aerosol therapy, has created the opportunity for new generation of anticholinergics. Okay. The third type of medication is corticosteroids, which treat the underlying inflammation of the lung that cause the disease, which asthma. And corticosteroids is associated with substantial systemic side effect. Okay, and aerosol advantage of requiring low doses sufficient to achieve a local therapeutic effect, but resulting in small circulating concentration of the capable of causing systemic side effect. Meaning the aerosol corticosteroids have a problem in terms of side effect if it has high concentration, and then the aerosol technology can deliver this type of drugs, okay, towards with good local therapeutic effect, but with less wind and a small amount of concentration that can reduce the systemic side effect. So that's why it's important, it advantages the use of aerosol in delivering corticosteroid. And then we have chromones. Okay, chromone use mast cell stabilization, which preventing the histamine release involved in local hypersensitivity of lungs. The major action of this compound is still obscure, but they have found a particular application in acid-induced asthma. Right. Next, we have emphysema. Okay, the most prominent example of drug for treatment of emphysema is A1 antitrypsin. So in emphysema, it the problem is related to this disease is it will reduce the free elasticity in the lungs, which preventing the structural remodeling of the lung, which lead to poor gases exchange and severe disability associated with the disease. So the problem is once it uh, become bigger, it, it it's difficult to reconstruct, okay, of the lungs. That's the problem that lead to the poor gases exchange. So the oxygens, okay, received in the blood in your body will be reduced. So that's the problem. And then we have cystic fibrosis, which imbalance in airways chloride ion concentration due to the absence of cystic fibrosis transmembrane regulator. So three approach have been taken, okay, to the use of aerosol in cystic fibrosis treatment. Delivery recombinant human DNAs to cleave the tangle of leukocyte DNA, and aerosol is employed to deliver an antibacterial agent, and nucleic acid is employed to correct the genetic imbalance in cystic fibrosis operation. So that's the delivery of cystic fibrosis. So, all right, other locality agents, we have amikacin, we have amphotericin, and we have pentamidine. All right. So basically, the common use of aerosol technology commercially available, and we can see a lot have been used by patient individually where they use to treatment asthma. Okay, the difficult in respiratory problem, the difficulty in breathing. So that's the most common application of the aerosol technology. So however, we see they also can be delivered other than type of breathing technique but related towards the lung problem. Okay, next, we will look at the formulation variables. So formulation variables were we look at the component of the formulation within the whole of these three system: PMDI, DPI and nebulizer to see the major component, the major substance or ingredients that within the system. All right. The compatibility with various solvent, propellant, alcohol, water, liquid, glycerol, for example, and solid. Okay, lactose for DPI. Base excipient is key to chemical and physical stability of the product. So we will look at the excipients, the interactions, and stability. So in DPI, we look at the excipient for PMDI. Okay, so it must be inert material in which most drug exhibit limited solubility. Two favorite approach into incorporating first surfactant to disperse respirable particle in suspension. We have CFC and then HFA. And then we add a co-solvent such as ethanol to bring the drug into the solution, which achieve a molecularly homogeneous distribution in the propellant. So it is important, again, not only for the aerosol-based system, but the homogeneous distribution must be achieved in all type of formulation. That's the main idea. That's the one of the main parameters in preparing the formulation, right? In the next, with suspension formulation are prepared to achieve control flocculation, which means ease of dispersion upon shaking. So physico-chemical phenomena that disrupt the stability of the product. However, the problem in PMDI were related to the moisture, all right, which first hydrolysis of molecule that will lead to degradation. The second one, interaction between particle that will lead to aggregation, and then the particle growth that change those delivery. So that's with the problem, because at the we, as I mentioned in the earlier topic, the particle size is very important because we have range of size that can be of drugs that can be delivered, can be achieved by that particular our lungs, right? So in dry powder system for excipients, prepared from pure drug substance and blend with lactose. Okay, lactose blend consists of respirable drug particle and large excipient particles, which mean the lactose. The excipient is included as a diluent to aid in dispersing the drug and fluidizing agent to assist the dispersion. Some spray dry particles has been prepared using other generally regarded as safe grass substances such as lecithin, human albumin, polylysine, and polyarginine. Okay, this is some addition excipients that will also been added to the DPI other than the drugs and other than the lactose to help the uh, in dispensing the drug and fluidizing the agent to assist the dispersion. So in in nebulizer solution, the excipient solution intended for delivery from the nebulizer are subject to the rules guiding general solution chemistry. Susceptibility to hydrolysis mitigates against solution formulation, and light and heat may accelerate degradation. This is related with the stability, right? The presence of complexing agent will influence solubility. Using concentration near the solubility limits of the drug may result in precipitation. So nebulizer solution are now prepared as a sterile product due to degradation of the drug. So the excipient must be at the end, it is important that at the end all the type of formulation, okay, all the ingredient must be have its own individual advantages towards our targeted system and targeted drugs and targeted application. And at the end, it is important to ensure that the recipient or other any ingredient that will be added into the system must be what we call must be integrated, must be stable and will not interfere with the therapeutic effects of our drugs towards the targeted applications. That's the most important thing. Okay, next we look at interaction. Okay, interaction in PMDIs, the incorporation of surfactant in PMDI contributes to suspension stability. Okay, doses throughout the lifespan of the product. Model system that side chain interaction may occur, which will ultimately influence the physicochemical properties of the product. This is the things that I mentioned, the interaction or reaction within the whole ingredients, and especially uh, towards the drugs that will reduce it efficiency of the therapeutic effect. Okay, right. In dry powder system, one approved excipients, the drug have to be compatible with lactose. because lactose is the main ingredients in DPI. Moisture absorption, chemical degradation by hydrolysis, or physical instability due to capillary forces is one of the interaction that related towards the dry powder system that will lead to it limitation in terms of delivering the drugs. Okay, hydrophobic effect become the source of aggregation and the stability of the excipient itself. In nebulizer solution, excipients such as sodium chloride achieve isotonicity with body fluids, but interaction occurred in which a precipitate of drug was formed, okay, with sodium chloride that been used, and particle of aggregate size in suspension may influence the size of the droplet derived from an air jet nebulizer. Again, right? So again, in the end, the idea is to ensure that all the ingredients will not affect the therapeutic effects. And in terms of aerosol technology, it is important to ensure that the problem is not related or will not affect, okay, the particle size, because the particle size is most important, one of the most important parameters that for us or pharmacists must to ensure in order to deliver drugs into the lungs, to ensure the therapeutic effects were in the best condition. Okay, in terms of stability, right? In terms of stability, these are potential for drug interact. So not only for the MPI, nebulizer, and DPI, all of these parameters or atmosphere, okay, or system will act with the stability of the 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 drugs, okay, such as solvent, excipient, packaging material, atmospheric exposure, oxygen, light, heat. So the drugs is not only will react the possibility reaction with in terms of solvent, in terms of excipient, which mean the ingredient within the formulation, but we must look at a little bit. Okay, we must take measure the interaction of the drugs within the packaging material, the outside atmosphere, the light itself, and the heat. Okay, because it is important to select the packaging material for shelf life, for efficiency, for stability, and also related to light and heat were transportation of our drugs, the mobility of our system. Either it can be in terms of long time use or short time use. So all of these some parameters must be take note, okay, in terms of stability of our system. This is not all uh, covering all type of stability or interaction with our drugs, but these some kind of major uh, preparation or parameters that must be take note. Okay, the performance of PMDIs and DPI is criticized in terms of efficiency and reproducibility of those delivery, particle size, distribution and the range of storage condition, and temperature and humidity. Okay, this is the terms, okay, in preparation of PMDI and DPI. Okay, because the efficiency and velocity of those delivery, particle size were related with how we were related with the mobility, okay, for the patient to be used the PMDI and DPI. So in order to ensure that the use of PMDI and DPI in terms of long shelf life, okay, it is important because at the end the stability will relate with the efficiency of that particular drugs, and that we relate what the particle size on how it be delivered. So that's the idea, right? So I think that's all. Thank you very much. I see you again in the next topic. Thank you very much. Bye.